Okay, and we're live. Dr. Duke and Dr. Cross, thank you so much for the presentation. It's really informative. I learned a lot about the work of breathing. And I was really surprised by some of your findings, particularly that people are using the wrong units. That seems almost crazy. I think you said something like 70% of people were reporting the wrong units. When I look at that kind of thing, I guess you sort of hope that that gets captured in peer review. Someone looks at the paper and says, well, you used the wrong units. But clearly that's not always happening. So I guess you referenced IRMC that I found out on Twitter, and you can find it on Google. So are you guys establishing guidelines there? If I were to publish a paper in this space, would I be able to go through like a checkbox and make sure I'm doing everything right?
Yeah, JJ, you want to take that?
What's that, Troy?
Did you want to take that one?
I can't hear you, but that's okay. I could try to answer.
Okay.
At present, there's not a sort of a checklist that one could follow to make sure they're doing everything the right way. Ultimately, we're hoping to build towards a sort of publication where one could follow and say, when trying to measure the work of breathing, here are some practices and principles that you should follow to do this in the most correct manner possible, to try to provide some guidelines for everybody to follow. There's tons of examples of this in physiology. Another thing that my lab is interested in is measuring blood volume, which is done using something called carbon monoxide rebreathing. It's almost as scary as it sounds. But there is a publication out there which is literally a guidebook for how to do this. How long, how big is the circuit? How much carbon monoxide do you inject? So on and so forth. That a student can pull up and say, this is what this should be like. These are the values I should get when I do this measurement appropriately. And that's what we're really trying to work towards. The systematic review, our survey findings getting out there is ultimately going to, I think, help us make the case for how much this needs to happen in this field of respiratory mechanics.
Yeah, I also want to mention, I have many more questions. I'm really interested in a lot of stuff you guys talked about, but if anyone else has questions, you can post them and we'll prioritize your questions. So just stick them in the Q&A box and I'll read them out loud. So along the same lines with those wrong units, I'm curious about the trends. Dr. Cross, you talked a lot about sort of the overall prevalence of different things, but are those things changing? Were all those methods you mentioned, I think the Headstrand, Otis, Campbell, were those all created at the same time? Or are people moving towards a specific method?
Can you hear me okay? Is my audio coming through?
I can hear you. I'm not
JJ
sure why.
can't. That's okay. It's not
strange.
The first time JJ ignores me. So. But as long as you can, then other people can. So, yeah, that was definitely one of the goals, was to look at over the decades, are there any trends in doing one method versus another? We don't, unfortunately, have that many publications across the decades since creation, which is really in this field since the 1940s, 1950s, because it's in the last 20 years has really been when we've seen a skyrocket in this type of literature. So to look at it back as far as the 1950s to see what's happening, sure, technology has changed. There are definitely some nuanced technologies out there that don't exist anymore or just gone out of fashion. Like the Bell spirometer or the wedge spirometer, which are these massive devices to measure volume. You just don't do that anymore during exercise, as we have smaller devices that can fit in the mouthpiece. So definitely looking at trends over time was a original goal. And I would say that there is definitely a bit more of a shift towards more convenient measures of the work of breathing. The pressure volume diagram, I think, is probably the most common across all time, but relative to the other more complicated measurements like the Campbell, I think that was definitely popular around the 60s and 70s when Campbell kind of published his PhD monograph on it, and it got taken up a little bit, relatively speaking. And then it just got more difficult for people to measure it, because I think the knowledge on how to construct these loops, and even having electronics and software and the ability to electronically store data, didn't really exist in a way that allowed people to do what we do, which is like use MATLAB now to ensemble average, signals recorded at 200 hertz. Like it was definitely probably falling out of favor because of how arduous it was to make those measurements. It's making a comeback. That Campbell diagram is making a comeback. And that's another thing we really hope to achieve in the next year or 2 years, is to create an open source mechanical analysis program or software package where people can do that. Because I think one of the biggest barriers that we're finding through anecdotal evidence is that people just don't want to do that because they don't know how, I think it scares most people off the method because there's a lot of programming in the background to get it right. And there is currently no widely available and open source software to do that easily.
So it looks like, Dr. Duke, you're kind of flashing in and out. Did that audio end up working for you a little bit, or still no luck? Can you hear me?
Not now. His audio doesn't work for me.
I can't I can't hear you any longer either, Dr. Duke. Let's see. I'm not sure how to help you. That sometimes works. You could try restarting it if you can hear us. But I'll keep going with a couple of questions and give you a little time to get it working again. So a question from the audience we had was about the different methods and which groups are using which methods. So are like the cardiovascular people using one method, and the physiology, the exercise people using a different method? Are there correlations there?
I think with the cardiovascular group, because there is so little in that field relative to healthies and pulmonary disease patients, there might be a handful of studies. And I think, one of them will be my study. We did the Campbell diagram. Pressure volume loops back in the 50s and 60s would be the other one. So I don't know if there's any trends between the groups that are using it, and rather the populations that it's being used on. I think personally interesting is that, and maybe we should try capturing this somehow, JJ, is even within a laboratory, the group of investigators from one paper to the next paper will use a different method of quantifying the work of breathing. And that might be going from the pressure volume to a head strand to a modified Campbell, and then in another series of papers, the pressure volume, and so on. And so not just between groups, but even within a group, there's a lack of consistency of going with one method or another. Maybe that's not a bad thing, because in one of our recent papers where we've compared what you get when you measure the work of breathing via the different 4 most popular methods, there are a lot of reasons that go into making your decision on which method to use. Some are related to the devices you have. Maybe you're trying to make a field measurement. Maybe you're collaborating in a different lab which doesn't have access to these things, or the experimental design doesn't allow you to make the measurements you want to do the more advanced procedures. So there is a lot of that goes on. But I think the thing that really we really want to champion is this transparency, that when you do choose a method, to try to explain or verbalize some reason why that method was chosen, or in the very least, acknowledge the limitations of that method. Because without doing that, we sort of breed this fallacy or this misconception that all measurements of the work of breathing are the same. Doesn't really matter. When I say work of breathing, you just believe me that it's the same thing, even though I do it 3 different ways in my prior publications. It's the same thing. If I don't go to that extra effort in my discussion section and say, this is what this measurement actually measures, so we can only comment on that, then I'm not doing what I can as a scientist to contextualize the scope of my findings. That's probably the major thing for us, is even within a group, and we're liable to it as well. A lot of this is a personal reflection on our own work as well, putting that in context and going, how good are we at doing these things too? So moving forward, we endeavor to do them better. But that was certainly something I found frustrating learning this field, is trying to get a sense of what is the right way to do this when even the one group does it differently all the time. So yeah, hopefully that answers your question.
Yeah, I think that and then some. Dr. Duke, I see him muted now. Maybe give him a couple more minutes. I hope he comes back. Any luck now?
Can you hear me?
Yes.
I can hear you. Yes.
Okay. I didn't hear anything that Troy said still, but I think I probably have a few guesses of what he said, and I think I'll just comment on TJ's question about the most frequent method being the least informative. It's also the easiest to do, and the computations, just the collection of all the data aside, the computation is so much easier to do, and from a computing standpoint, like on the computer coding, it's just far easier to do than any of the other methods. And so most just, I think, revert to that. And that's why that is the most commonly used method.
I have a really basic question about that, actually. Maybe I'm misunderstanding something, but from your presentation and looking at some of your papers, it sort of seemed like the way this works is that there's that PV, area that you end up generating, and then there are a few methods of calculating, portions of that area. At least that's one way it breaks down. So when people are actually reporting the data, do they share those those images that are getting generated, or is there some way of sharing the raw data that those different methods are actually getting calculated off of?
Yeah, I think to varying extents, well, I would say that a pretty broad answer to that question would be no. When papers are published, if any of the raw data is presented or made accessible freely, I think it would be the values that are obtained from those pressure volume diagrams, rather than the actual pressure volume diagrams themselves. So there's definitely that feeling of smoke and mirrors, like there is that, what you might present a representative pressure volume diagram, which is usually, a pretty good one of the lot, because you can get some real ugly looking ones. Doesn't make them wrong. They're just not pretty to look at. And they might not get across the schematic, representation of the abstract thoughts that you want someone to gain from your, more smooth looking pressure volume diagram. So no, I don't think it's often the case that a pressure volume diagram in its raw form is given. And that's probably a reflection on how variable it is out there at the moment. To arrive at that one conclusion, you might have someone who's written code in LabVIEW and generated these values that they've paid an engineer to do that software for them, but they themselves can't even get access to that raw data. They just get the processed outputs.
Yeah.
Whereas, I've written this code in MATLAB, in R, and LabVIEW, and in each time, just handling that data is a little bit differently. The outputs are a bit different. So maybe that's a symptom of a lack of a standardized software out there to do this analysis, which if we could get that and have a export function that was standard across all studies, it would become more common, I would say, to provide these raw data and loops as additional supplement attachments to publications in the future.
I'm a little surprised that the device manufacturers don't have like a plugin to like an open source package, or sort of like a some sort of recommended way for that to work already. I see both of you nodding. Do they do that a little bit? Dr. Duke?
I think the primary manufacturer of the balloon, like the air-filled balloon catheter, it's not designed to study exercise. That wasn't like the impetus for the design of that device. It was for a clinical situation where a patient was being ventilated. And so then the respiratory and exercise physiologists were like, oh, I'm gonna use that. I'm gonna use that during exercise, and kind of off we've gone. They're very easy to get. They're relatively inexpensive to get as well, and making them yourself is an option, but it also requires a bit of technical ability. Only when I've had 6 cups of coffee can my hands be steady enough to like assemble a catheter
Wow.
from scratch. And so the guidelines from the manufacturer are like, oh yeah, sure, it's fine. And that's kind of that, because it's really not. We're applying it in a way that it wasn't really intended.
Yeah, there is one commercial company out there that does offer a bit of like a full package, which is quite expensive. And what I've taken to over the years is to build my own equipment, and because of that, we're able to generate lower cost alternatives. But the increased cost with that is more the complexity and time developing the software to use the raw equipment itself. But yeah, that in the clinical sphere, there are ventilators that you plug a balloon into and it'll give you worker breathing measurements. There's even ventilators out there that will adjust the proportional assisted, the assisted ventilation given someone's own work of breathing as well. But there's nothing about helping us do that during exercise when someone's honking away at like 200 liters per minute going in and out of the lungs. It's really difficult to get that. So it's a lot of bespoke equipment and a lot of bespoke software.
Yeah, maybe kind of a naive question, but I'm curious about calibration in general. If you're sort of calibrating against the tool, it seems like there might be opportunities to bring in other metrics. So, for instance, if cycling is the main thing people are using, then you could use the output of the bike, right? Like how many watts is someone generating? I assume that's related to the work of breathing. Maybe some people are more efficient than other people, but it would provide a calibration opportunity. Is that kind of thing done at all?
JJ, did you want to chime in on that? No, probably can't hear me. That's okay. I'll do that. So yeah, I guess that's like a biocal. Like you're using something that you should know in physiology evokes a specific response, and we do that with gas exchange equipment. So measuring oxygen consumed and carbon dioxide produced at the mouth. You could quite easily get almost anybody in the room on a bike exercising at 100 watts, and generally speaking, their oxygen uptake will be 1.5 liters per minute. That's your rule of thumb. But when it comes to the work of breathing, because everyone's lungs are different sizes, you've got different heights, you've got different body masses, there really isn't a standard prediction equation or a rule of thumb where you could say this measurement of the work of breathing is exactly what I'd expect for that workload. To sort of like as a sanity check, you really can't do that. All you can really do is say, is this method or is this measurement of the work of breathing for this work rate nonsensical? That's probably the highest extent that you could go to using a sanity check there against external tools like a work rate on a bike. But even then, you've got to temper that with the fact that maybe for that person, the work of breathing just is generally high, and it's higher than the average individual. So long as it goes up when you increase your breathing rate, and it trends in that direction, I think your measurements are sound, but definitely, yeah, it's really difficult because the work of breathing measured by different methods as well, that changes entirely too. Yeah.
So JJ, maybe I'll start with you for the next couple questions, and then move to Troy afterwards. That way,
I think Airmeet is plotting against me and Troy because it must know that we just waffle about and we'll just keep talking and talking. So it's trying to minimize our ability to, or at least my ability to talk to him and hear what he's.
Yeah. We might move to Zoom or something in the future. But I'm actually just curious, maybe a little bit away from the direct research, but about the review experience. Have you done this before? And what would you do differently if you were to start the whole process over again?
I have not done a systematic review before. Admittedly, I didn't see what I could gain from it until I started doing it. And then I realized just how much I could gain from it. the most simplest and basic thing is having a place where all of the literature that measures the thing that I might be interested in measuring is all like in one area. And I have referred so many students to, there's a bunch of different ways to measure chest wall compliance. Ask Colin, and he can give you all of the papers that we
Wow.
checked yes on.
That's cool. Yeah.
I don't know what I would do different. I think Troy, who's really been the leader of this effort, really did a great job of thinking not about like what do we want to know right now, but what later might we want to get out of the literature, so to speak. And so that's why we had 50 labels, because we wanted to go through this and do all of the work on the front end and not have to go, oh, well, now we want to know whether they calibrated or not. We didn't think of that. And so we went back and we would have to go all the way through all of the literature again. I guess I would have probably brought other students in to be involved in the data extraction piece, because I know Colin, who was involved in the very, very beginning, really learned a tremendous amount about the different devices. And he and I had so many conversations like, well, how does this one work? And I'd be like, I think this is how it works, but I'm actually not 100% sure. And so we all learned a lot about all of the different ways things are measured. So I can't fathom that I would have done it differently, but I'm really glad that we went through this. And I think there's a lot of opportunities to do this kind of thing again.
Troy, did you want to add to that at all?
No, he said exactly what I was going to say. Yep. I don't see much that we would do differently, and it was an awesome learning experience. Absolutely.
JJ, I wonder that comment you made about sort of being able to filter to a set of articles and give that to a student, that resonates with me a lot. We've had to learn some new fields when we've done these projects, and it's amazing sort of by structuring the data, just by answering simple questions, sort of changes the way you think about things. But that resource that you've created is like a pretty valuable curation for you now then.
Yes. Yes.
Do you think that like, would you want to keep adding to that? Or, if you created a new project, now you have different projects to give to a student, and that seems just like could get confusing pretty quickly. Have you thought about that at all?
I think I think it can definitely get confusing. And my first thought in answering your question was thinking about other sub areas within, respiratory mechanics that I might be interested in sort of collating all of the literature, if you will. And one example that popped into my mind was Colin, one of the students working on the project, is interested in respiratory muscle fatigue. And so like having that be a sub systematic review with the real objective being, let's get all of the papers that have measured this parameter. And so I could see the confusing part being all of the overlap between the repository we've created with this systematic review and the repository that he would create in that hypothetical situation. But I certainly see it as a valuable tool, really, to really have an organized way to search the literature for students doing theses projects, because your review of literature in your thesis and dissertation does have to be comprehensive. You do need to kind of collate all of the available information to accurately and adequately provide a review of the pertinent literature and how your new study is going to fit in with that. So I just think it's a valuable tool that I could see being using myself in the future.
Yeah, the sort of the concept of having your review be a database that every student can add to over time, or every time you do a new project, you're sort of adding value to that curation, is one that we think about a lot. And we don't have the answer right now, but it's something we're absolutely working towards. On that concept, actually, Troy, I think when I saw your figures, I think they looked like they were generated maybe with ggplot or an R package. So if you're using R a lot, are you aware that there's a plugin with that on Sysrev, or did you use that? I'm not sure.
No, I didn't actually, but that would be quite useful. Is that like access to the API? So I give it a project name, it pulls then the results from that.
That's it. Yep. So
Okay.
you couple lines of code, you can pull all the data down from your project. We'll
I guess,
make sure we show you how to do that.
yeah, because that would facilitate the whole idea behind a living review, right? Is that I can just rerun the code rather than download, organize, put the code into R, just by literally having a pull request for the data, then it runs and updates. That'd be, yeah, that sounds like a cool little workflow. And yep, definitely it was ggplot. Those are pretty obvious aesthetics. And then, yeah, I guess to augment what JJ was saying too, and to answer your question, Tom, is that moving forward, I think this is a really difficult systematic review to be a living review, to be one that updates, only because we progressively see more and more studies added within a given time frame, but it takes so long to get all persons involved in the data extraction on the same page to then annotate the correct labels. And so it's not as, I guess, more simple or straightforward as checkboxes or a whole bunch of Booleans where you can go yes, no, yes, no, yes, no, yes, no. So the investment in training up new investigators to do that, I think, discourages us in a sense from making it a living review, more so than once it's published, we kind of like stock it, lock it, and refer to it. Whereas I definitely do see a value in expanding it over the years, but it's more so the time it takes at this current stage to label it and annotate each article that I think is a bit more discouraging to keep that process ongoing.
Yeah, certainly with 50 labels, it takes a lot of time to look at an article and extract everything. We had a large project recently that, every paper really took like 4 hours or so for someone to sit there and look at it and, understand it well enough, So it's definitely can be challenging. One thought around that is if you could do pieces of it at a time, right? Like if you could have someone answer only a couple of questions or do other kinds of workflows. Did you JJ, did your group experiment at all with like different ways of handling the work, or did everyone just kind of invent their own way and click the buttons and get it done?
When we proceeded to each new phase or stage of the project, we would have a group meeting, and we would, Troy and I would kind of work through this. Is, first Troy and I would have a conversation, then we would have the whole group and say, this is how we envision this going. What does everyone think? Does everyone have questions? Does anyone have any comments? And then we would kind of like send them off on the loose. And as things came up, we would kind of have a group email conversation, and if needed, another meeting. We had, if I remember correctly, we had a conversation about going through and answering question 1 for every paper, like what's the population and how many participants are being studied, how many boys, how many girls, etc. And I think what we ultimately landed on, and why we didn't do it that way, is so that one person could work through an entire paper rather than have me work through 10 of this thing and then another person the next 10 through those 10 papers. Just from having the same sort of approach to how the person would kind of extract the information and the types of terms they would use in the free text areas of the program. And it seemed to work pretty well to doing it that way. And I think once you get into the nitty gritty of a single paper, extracting more information becomes less of a task.
You get more efficient. It's like a bigger,
right. Like that inertia to kind of open up that paper and start on it is the hardest thing to overcome. And then kind of once you're going, you really can kind of get to the data. And sometimes I never did them in order, except for maybe I always did the first one first because it was always kind of the first thing I came to. And then I would just kind of scroll and look for key terms, and then I might answer some in the middle and then jump around to the bottom and then kind of come back up and then make sure I had something for every label. And we certainly talked a lot with the students about the process and working through some of it with them. Kind of like one-on-one, we would sit down and go through a couple together and then send them on their own to try and suss it out themselves.
Troy, any comment from you on that?
The only thing I guess I could say is that as we moved through, what sort of facilitated that streamlining within a single article for one person to do all of it was that I think by the third stage, where we were doing the full text data extraction, I'd then come to realize the functionality behind the text input, where you could put regular expressions in to limit what people actually would say. So I think once we figured that out, it reduced the ambiguity around having our students sort of like, how should I type this in? How should I type? There would only be a specific set of ways that they could type in pneumatic or turbine, so we had little codes and regular expressions. And we sort of kept it that way because it allowed us to, rather than having an enumerated drop-down box, it allowed us to have the freedom of having our ability, I guess, to have other bits and pieces encoded inside of a response that we might be able to extract out later, like age, where we wanted to make sure it was all alphanumeric, but you would have the mean, and then in brackets you would have the standard deviation, and then post-processing in R, I could use pattern recognition to get out what was the age, what was the mean, because otherwise you end up with so many labels just for the mean, just for the standard deviation, and so on. Another thing we found quite useful to help that process along is a lot of studies had multiple subject groups. They might have had a healthy and a heart failure group, or a healthy and a trained group. And when we wanted to calculate sample sizes, we didn't know a priori how many groups each study would have. So what we used with the text input was the ability to have multiple responses, so that when we put it into R later, we could partial them out or separate those responses and know that, well, the first response was for the healthy, and this is the age, and this is the sex, and so on. And as long as we kept that constant with the multi-line inputs or the multiple inputs, then things would marry up with each other. And once we figured that out too, that sort of sped us along forward to the data extraction pathway. So that was really helpful.
Super happy you got a lot out of those regular expressions. We use those a lot as well. And they do match really well with the code. So, if you know the pattern looks like this, then you can extract it like that. Maybe a final question is about the downstream impacts of this research. So just a couple basic sub questions are you going to make the data available in your publication from your review? And how do you expect people to respond? I mean, is this going to impact a lot of the research down the line?
I'll answer the second question first, and then let Troy handle the first question. I hope that the results are taken for what they are. They're the reality of what the literature looks like. This isn't an accusation or an attack on the field per se. It's just about us trying to gather the information and kind of put it all in one place. This is what is in the literature, and I hope people, appreciative sounds a little bit self-indulgent, but I hope people see the value in that we have taken the time to collect all of this information and put it in one place, and that it will more readily bring forward the fact that it is so difficult to go into this literature and do this method. So difficult. And so I hope that it will usher in a little more transparency and clarity, and more of a push from the author's side to include details in the methods section of papers, because anybody in this area, anybody in physiology, you submit a paper and the first comment from every reviewer is, this paper is way too long. And the methods are always the easiest thing to chop, because you can go as before, as before, as before. And so we're hoping that this will really bring some awareness and really get the authors to say, because it's not always the author's fault. It's always the reviewer's fault. And it's always reviewer 3. They're always the one that's messing everything up for everyone. That's a little scientific review joke, but hopefully this will give the ammunition for authors to say, no, this is really important that we include this information, even though it is redundant from the previous 5 papers that we have published. It is really important that it is included in all of our publications.
And I guess to answer the first question, second, is that when this publication gets out there, it is our intention to make sure that there's enough raw data there for people to be able to use this as a database themselves, to be able to determine, okay, well, if I want to see, and like you had mentioned, Tom, if I want to filter by a specific method of the work of breathing, so I can go to those papers and read how they may have described it and understand things, then that could be easy enough done even just in an Excel spreadsheet using the filter function. So we'd want to be able to produce as like a product, not just the summary, but having that as a supplement to the publication, that data base of literature.
Yeah, just I know I said I'd wrap it, but just if you're going to distribute the data, something we've been thinking about is all these people put in tremendous amount of effort. It's really hard to understate or overstate the amount of work you put into this project. It's tremendous. And, there's 2.5 million, 3 million labels have been assigned to articles on Sysrev now. So there's a lot of this kind of effort going on. And it would be useful for the next group that wants to do this project if they didn't have to redo everything you did. So if there were a way that someone could take your project and just, take all the data and import it into their own review, is that something you think researchers would benefit from? Too much complexity there? Or do you see a way for that kind of idea to work?
Yeah, I mean, I think that would definitely... I mean, it's sort of the way forward with systematic reviews too, right? Like the new PRISMA guidelines from 2020 now require you to identify whether or not you are piggybacking off of existing reviews in their PRISMA flowchart as well. So I think, it's coming forward more readily that, yeah, if someone's already done this, it would be good to have a standardized mechanism to say, okay, I'll take it from here, with more data or in a slightly different way. I wonder if the complexities might be more, not so much in the included articles, but maybe in the labels themselves, given that maybe slightly different data would warrant extraction between the projects. And the added complexity too, that perhaps that search strategy might have a nuance to it, that would limit the scope to like 90% of the articles you really wanted, but it's at least a good start to complement it, which I'm not sure if Sysrev offers this for, because most of these projects are public. Are public members or members of the public able to go to a project and download the XML of the references that were included? Is that data available for
public projects? You can download the sources that were used. That's correct.
Okay.
You cannot download PDFs, but
okay.
The other sources are available.
Yeah.
Okay. So I have a lot more to ask, but I think I want to be respectful of your time. And thank you, JJ. Thank you, Troy. This was really fantastic today. We're huge fans of your work, and we hope we see many more projects to come. So thank you both very much.
Thanks, Tom.
Thank you for having us.